Oh, I see the number on the attendee list is going up. Here it goes. Wow, it's really zooming. Welcome everyone as you join. So I'm Sue Fletcher-Watson. I'm a psychologist and I'm based in the Division of Psychiatry. And I'm not going to fully introduce our speaker yet, but we're going to hear from Professor Chiara Nosarti today. And I'll do a full introduction in a couple of minutes, but I think we're just going to hold for a minute or two to allow everyone to kind of log in and make sure that we've got all of the attendees with us before we get started. So, so you can just make yourselves comfortable for a couple of moments. Okay, I'm back again. So I can see that the number on the attendee list has stabilized. So I'm going to assume that that means that everyone who wants to be here is here. So yeah, it gives me absolute huge pleasure to welcome Chiara Nosarti to come and deliver our remote special lecture this week. Chiara is a professor of neurodevelopmental and mental health at King's College London in the Department of Child and Adolescent Psychiatry. And like me, she is a psychologist, neuropsychologist who has infiltrated psychiatry and has been working on understanding consequences of preterm birth now for 2 decades. So we're really fortunate to have her expertise, and I absolutely know that this is going to be a fascinating lecture, the title of which is The Legacy of Preterm Birth on Brain Structure and Function in Adulthood. So I'll hand over to you, Chiara, without further ado, and I'll just remind everyone that you can post questions in the Q&A section of the Zoom webinar, or if you can't find that, then in the chat. You can post them as you go along. We will ignore them until the end. And then what we'll do at the end is I'll turn my video and my microphone back on, and I'll look through those Q&A questions, and I will pose questions to Chiara based on what I think are the kind of themes that are coming out in what you're asking. So please do stick around for the Q&A, and I'll do my best to get through as many of your questions as we can before we run out of time. Okay, over to you, Chiara. Thank you.
Thank you, Sue. It's great to be here with you in Edinburgh, although virtually this time. So what I'd like to do, I'd like to start just sort of highlighting, why is it important to study preterm birth. Preterm birth is defined by the World Health Organization as birth at less than 37 weeks of gestation, and it represents between 5 and 13% of all live births, depending sort of geographic location. Very preterm birth is defined as birth equal or less than 32 weeks of gestation and represents about 1% of all live births. Thanks to advances in neonatal intensive care, more preterm babies are now surviving, and also the incidence of sort of serious neurological problems is decreasing, and only between 5 and 10% are affected with cerebral palsy, for instance. Unfortunately, cognitive and behavioral problems do persist in childhood and later in life, and these are partly due to alterations in neurodevelopment. And this is what the main body of my talk will relate to today. So in these sessions, I'll give you an overview of results of studies that my colleagues and I at King's College London have conducted in adulthood. I'd like to talk about possible mechanisms associated with adverse cognitive outcomes and increased psychopathology. And I'd like also to perhaps think about future lines of research in conclusion. Again, I think in terms of understanding the neurodevelopmental patterns associated with very preterm birth, we very much need to consider what happens in terms of brain development during the third trimester of gestation. And this is the time during which the majority of preterm children are born. So this is a very critical phase of development of rapid brain growth, and which includes enormously sort of critical processes of differentiation and maturation of pre-oligodendrocytes, formation of synapses between thalamocortical afferents and sublet neurons, axonal growth, and cortical gyrification. If I'd like to kind of draw your attention to this diagram, this is the brain in coronal sections at 28 weeks of gestation, and shows these sort of critical events in cortical development. So in A, we see the axons in green, which emanate from the thalamus, the corpus callosum, and initially on the sublet neurons. And then here in B, we see the proliferation and migration of GABAergic interneurons from the subventricular zone here in blue, and the general matrix to the cortex and to the thalamus. But what is important is that already by the time of like term equivalent, by 40 weeks age equivalent, there are very substantial differences in the way the preterm brain and the term brain is connected. This is a study done a few years ago by Gareth Ball and colleagues. They were then at Imperial College looking at thalamocortical connectivity. And these areas in yellow here represent regions of significantly lower thalamocortical connectivity in preterm infants compared to controls. And can you see they're really, really sort of like extensive and widespread. And what is important is that this impaired connectivity or alter connectivity is specifically located in regions that are believed to be important for a wide range of both cognitive and behavioral functions, and may be in part responsible for the development of or the exacerbation of problems as preterm individuals grow older. For, I think now it's already about 20 years, I've been involved in the University College London Hospital follow-up study. This is a large study that was initiated in the late 1970s and comprised all children born at less than 33 weeks of gestation at UCLH, and then they were admitted to the neonatal unit and entered a long-term follow-up. So neurodevelopmental assessments were conducted at age 1, 4, 8, 14, and we started with imaging, 18, and the last assessment was at the age of 30 and finished a few years ago. We really kind of administer a very large battery of assessments, including sort of a variety of neuropsychological, assessing neuropsychological functions, executive function, IQ, language processing, visual spatial processing, and so on. I'll talk a little bit about emotion processing in this section. And then we also looked at psychopathology and social functioning using a variety of instruments and 2 hours of imaging with a break. We did 1 hour and another hour. So what about the profile of a very preterm adult? So this is a summary of results at the age of 30. The study was led by Jasmine Crow. We used this neurocognitive battery, which was sort of like mainly assessing different aspects of executive processing. We also measured a variety of real-life achievements, both interviewer-based and self-rated. And these are the sort of details of the sample, just nothing remarkable. In terms of neuropsychological outcomes, you can see that here that basically the preterm adults had lower scores compared to controls in all functions assessed except this continuous performance test. They also had lower or poorer real life achievements. So they spent fewer years in full-time education. They were less likely to be in employment. But in terms of income, there were no differences from term-born controls. There were no differences in the relationship status. And this is sort of somehow a surprising finding. They were more likely to be biological parents compared to term-born controls. They had a lower global role function index score, but in the self-assessment, they didn't report to experience a significantly greater social adjustment problems, and they rated their kind of overall functioning as satisfactory. When we looked for association between the cognitive and outcomes and the real life achievements, we saw a positive association only in the preterm group here, the line in red. So to conclude, cognitive deficits persist into adult life following very preterm birth and may be partly accountable for lower levels of real life achievements seen in very preterm survivors. Of course, further studies are required to better understand how specific factors may influence outcomes. And this type of results also very much highlight the importance of the development of sort of remediation programs to be delivered at younger vulnerable group. And there's been sort of like mixed results in terms of success with specific target interventions, and the majority of studies has looked at working memory. What about sort of brain development, or you say sort of differences in sort of brain morphology by adult life? So this is a study led by Slava Karolis, who's now moved to Oxford. So we're looking at brain volumes at 20 years of age, so at the previous assessment. And I don't really want to go through this, but I just want to kind of demonstrate the extent of differences that are still persisting when an individual becomes an adult. And these areas in orange represent areas of decreased gray matter volume in the preterm adults compared to controls. But this particular study, the Slava study, was very much wanted to probe the aging process. And I know these individuals are only 30 years of age, but we wanted to see whether we could detect already at this relatively young age sort of specific patterns that may be indicative of accelerated aging. And we based this study on this interesting idea, which is referred to as the retrogenesis model, or last in, first out hypothesis. And you see this like U-shaped curve. And the idea here is that the areas of the brain that develop last are also the ones that are more likely to show the first, aging signs of the aging process. And another observation, again, from this really like a lovely paper was published a few years ago, what is showing that these areas that are showing these retrogenesis effects, they're also very much overlapping with areas of kind of volumetric differences that are often observed both in schizophrenia and in Alzheimer's disease. So what we did, we looked at all our sort of images collected from the age of 14 to the age of 40. And then we took publicly available data, 1,270 scans from sort of typically developing individual aged between 9 and 65 years. And these are just a sort of breakdown of our sample. And what we looked at differences in maturational index between the preterm individuals and controls. And this is refers to the correlation between predicted age and chronological age, which was taken as a measure of prediction accuracy. So if you think this 0 is the controls, we can see that at the global level and at the level of sort of brain modules, the preterm individuals showed a higher maturational index compared to controls, which reflects the idea that there might be patterns of accelerated brain aging. But also what is interesting is that other areas which are centrally implicated in a variety of sort of mental health or psychiatric disorders, such as the caudate nucleus, actually shows a relatively younger age in the preterm sample compared to controls. So to conclude, this study is showing that very preterm individuals do show accelerated brain maturation compared to controls. And this is, of course, important for future study of the aging process in very preterm samples. And we need to really understand whether these accelerated aging patterns are associated with real life implications. For instance, early cognitive decline as it observed in traumatic brain injury. Moving on, another study looking at whole brain connectivity, again, a study led by Slava Karolis. Here we looked at structural connectivity with respect to the brain network ability to support two generic tasks. So the processing of information within dedicated subnetworks or modules and exchange of information globally. And we use a graph theory approach, which I'm sure many of you are familiar with. The idea is that we're using cortical, subcortical, gray matter regions as nodes and white matter connections between regions as edges. So we focus on two network characteristics. One is modularity, which is the network capacity to organize into tightly interconnected clusters. This is an example of high modularity and low modularity. And the Rich Club, which is a measure for network capacity to pass information globally between segregated modules, and refers to nodes that are connected to one another to a greater extent than they are connected to the rest of the network. This is an example of the Rich Club. So results are showing us that they showed that there was a stronger Rich Club architecture in the preterm adults compared to controls. And this was despite a relatively lower quantity of white matter streamlines. But then the idea was, if the Rich Club is stronger, is this more stable or is it less stable to perturbation? So Slava had this lovely idea of creating a simulated lesion, and in order to establish the impact on individual elements on the whole network architecture. And so what we did two types of lesion. We lesioned each node individually and a set of nodes, and each tract individually or a set of tracts. And what we're showing is the results showing that lesioning a region or a set of connections did not produce any changes to modularity. And this actually is an error. It's a modularity, but it did so in the Rich Club index. So if you lesioned a set of connections, and specifically these connections, which are, see if it works, it's striatal cortical connections, and between the right hippocampus and the thalamus, the right putamen and the caudate nucleus, there was a greater drop in the Rich Club index in the preterm adults compared to controls. What was also interesting with these results is that there were associations between this index and these changes in Rich Club index and outcome measures, specifically with verbal IQ and with different aspects of executive functions, response inhibition, and information flow and rule learning. So this study in conclusion shows that preterm individuals show an altered role of frontal striatal connections in supporting a global exchange of information throughout the brain, and these network alterations correlate with measures of information flow and rule learning. Another approach that we've very much taken in this sample is looking at functional neural alterations, but taking into account possible structural alterations that we know are present in sort of atypically neurodeveloping individuals. And this is a study led by shown through this Walsh, who's now in the States. And what he did, he looked at working memory fMRI in relation and in combination with tractography. And one thing that I think was interesting to investigate was to subdivide the preterm group in terms of the extent of neonatal brain injury that they sustained. And this was based on neonatal ultrasound classification. So we classified perinatal brain injury as grade 3 and 4 intraventricular hemorrhage and ventricular dilatation. And we use a very sort of like typical and back task that you might be familiar with, like from 0 to 3 back. We also used a spherical deconvolution approach to allow for the estimation of multiple fiber directions within a single voxel. So you see, this is like the difference. This is a spherical deconvolution. You're looking at different directions within a single voxel, whereas this is a single fiber DTI. So in native space, we virtually dissected 2 tracts: the dorsal cingulum, which connects the middle frontal and parietal working memory structures, and the ventral cingulum, here in yellow, which connects sort of the parahippocampal region to the posterior cingulate and medioparietal regions. So in terms of behavioral results in scanner, there were no differences between our 3 groups. So it was like injury preterm, no injury preterm, and controls. So every group performed worse in the most difficult condition of the task. And these are the results of the fMRI study. So what I'm showing here in orange are the areas where preterm adults show decreased hemodynamic response compared to controls, and here the area in blue where they showed increased. But when we went to look at sort of activation, the time series for the 3 different groups, I think what was interesting to notice that these differences were driven by this group, so the group with perinatal brain injury. We also went to look at structural function relationships, and in terms of size and of the dorsal cingulum, the injury preterm group had a lower volume compared to the other 2 groups. And also there was a negative association between hemodynamic responses in this sort of perisylvia area and the size of the dorsal cingulum, again, in the PBI group only. So the smaller the dorsal cingulum, the greater the activation in the perisylvian cortex. Whereas these associations were not observed for the ventral cingulum. And then we really kind of thought, is this kind of unexpected non-working memory activation compensatory or indeed maladaptive? So what we did, we correlated the COPEs in the perisylvian cortex with the task performance in the most taxing task condition, so the 3 back. And notice again that there was association, positive association in the PBI group only. So the greater the activation in the perisylvian cortex, the better the task performance. So why this might be the case? We think that perhaps PBI individuals may learn to increase their use of the phonological loop to overcome early life working memory deficits. So it's a kind of like alternative strategy to ensure a successful task completion. Also, microstructural white matter damage to dorsal cingulum was associated with functional reorganization of the working memory system, although this is sort of like a cross-sectional study, so we are unable to ascertain when exactly during development these, I think perhaps these adaptive changes took place. Of course, structure-function relationship must be made with caution because this is a group with altered neuroanatomy and may have altered structure-function relationships. The next study I'd like to mention is actually was the first study of resting state functional connectivity in preterm adults, and this was led by Thomas White. We know like why functional connectivity. Functional connectivity relates to temporal dependence of neural activity patterns of anatomical separated brain regions, and examining the human brain as an integrative network of functionally integrating regions can provide new insights into large-scale neurocommunication. And of course, the complex cognition and the optimal cognitive integration, we know now that arises from activity in widely distributed networks. And we selected some networks based on the literature and based on the sort of cognitive deficits and behavioral problems that we noticed in preterm samples. So we looked at the frontal and the posterior default mode network, the left and right central executive network, and the striatal salience and insular salience networks. And why these networks? Because we know that intrinsic connectivity between these specific networks are believed to have a strong influence on information processing, and deficits in access, engagement, and disengagement may play a crucial role in psychopathology. And what we did here, we looked at functional connectivity, looking at kind of temporal relationship between activations in the different networks. And specifically, we used the Granger causality analysis, which refers to a temporal causality between time series that can be inferred from the extent to which previous values of one time series improve the ability to predict present values of another. I know there are a lot of methodological perhaps concerns with this method, but we felt it was very important to understand the direction of these associations. So the idea is, if you have two time series, 1 and B, if past value of A and B can predict the future value of B better than B alone, then time series A is believed to cause time series B. And what we found that there were differences between preterm adults, very preterm adult and controls, and it was decreased connectivity between the striatal salience network and the posterior default mode network predominantly, but also differences reciprocal between the frontal default mode network and the right central executive, and between the frontal DMN and the insular salience network. So to conclude, resting state functional connectivity of preterm born individuals does remain compromised in adulthood and may provide a mechanistic explanation for previous observed deficits in switching and executive function. But I think what is of note here is that the striatal salience network seem to be preferentially affected, suggesting that this region is a primary site of enduring effects of preterm birth in adulthood, and perhaps implications for increased risk of psychopathology. Another study looking at resting state functional connectivity looked at emotion processing. And this study was prompted by the observation that up to almost a third of very preterm children experience impairments in emotion regulation. And we know that emotion dysregulation represents a transdiagnostic factor that increases the risk of various types of psychopathology. And we are actually now sort of really using this model to look at psychopathology in much younger groups. So what we did here, we used an emotion recognition task, so offline. And so there were 6 universal emotions, something like that, at different levels of intensity. We used a seed-based analysis, resting state functional connectivity, so divided the amygdala into 3 subregions, and used the CON toolbox for the analysis. So at the behavioral level, we noticed that the preterm adults were less able, they were worse at recognizing anger than controls, but only at the low intensity. So when actually the recognition of the facial expression is really quite ambiguous, and they also took longer to do so. So looking at the results, we've seen here that the preterm adults show decreased connectivity between the left superficial amygdala and the posterior cingulate cortex and the precuneus compared to controls. And perhaps the amygdala activation during processing of social emotionally relevant stimuli may excessively suppress the activity of default mode brain regions, so the posterior cingulate and the precuneus, that are responsible for the cognitive evaluation of a given situation. This, of course, is our interpretation. We also noticed an increased amygdala STS connectivity, and we interpreted this as possibly representing a compensatory strategy in a suboptimally efficient emotional network. So in order to facilitate an emotional recognition by means of stronger integration between decoding of facial characteristics by the temporal areas and emotional salience by the amygdala. Something else that is like apparent as well, is not very kind of clear in this plot, but is that there was an association between the anger recognition score and the superficial amygdala to posterior cingulate connectivity only in the preterm group. Sorry, I lost the cursor. That's it. That's in red, and it was not the case in controls. Whereas I actually think this may be a sort of power issue because the line seems quite similar. Anyway, so the way these results were interpreted as deficits in recognizing anger may result from an inadequate amygdala modulation in preterm-born participants, and the difficulties in coordinating activity between systems responsible for processing emotion and those responsible for attention modulation. And if you think, of course, the posterior cingulate cortex is a very kind of important connector hub in cortical subcortical networks. And that takes us back to sort of like the previous results. So to conclude, very preterm adults display functional alterations in brain circuits that are critical to emotion processing. Of course, we need to further investigate how emotion processing may interact with other variables and contribute to the increased vulnerability to psychiatric disorders in very preterm individuals. But so far, I haven't really talked about preterm birth and psychopathology. And I'd just like to give you a couple of slides to summarize what we know. We know that by the age of 18, approximately 25% of very preterm adolescents will have a psychiatric diagnosis. And this is mainly ADHD, autism spectrum disorder, anxiety and depression, and emotional disorders. But they're also characterized by what Johnson and Marlowe defined as the preterm behavioral phenotype, which is characterized by sort of subthreshold levels of inattention and hyperactivity, social and emotional difficulties, and a greater risk for internalizing rather than externalizing problems. A recent meta-analysis looking at preterm adolescents and young adults found that they are 3.5 times more likely to be diagnosed with any psychiatric disorder compared to controls. And when looking at anxiety and depression, we have odd ratios of 2.86. Looking at sort of the most severe cases of psychiatric disorder with hospitalizations, there's also an increased risk if you are born very preterm or preterm, in fact. And this is a study that I had the fortune to conduct in collaboration with the Karolinska Institute in Sweden some years ago. And we looked at the population-based registered of 1,300,000 individuals, and really looked at hospitalization with individual diagnosis. And this is like sort of summary results, reference term birth. So I think what is interesting here is to see that there is a dose-dependent relationships. So the increased risk is for both those born at equal or less than 32 weeks, but also between 32 and 36 weeks. So the odd ratio is 1.6 here, 2.5, and so on. But basically the risk is increased across the board for psychosis, bipolar disorder, depression, eating disorder, and no significant effects for alcohol and drugs. Now, what about the University College London hospital sample? These are the results of our assessment at the age of 30. And here, instead of looking at specific diagnosis, we use sort of a more dimensional approach to look at psychopathology. At previous assessments, so a mean age of 18 years, 12% of preterm versus 5% of controls received a psychiatric diagnosis according to the clinical interview schedule. And the three most common diagnoses were mixed anxiety and depression, depressive episode, and agoraphobia. So to evaluate psychopathology, we use the comprehensive assessment of risk mental states, which is an assessment of predominantly psychotic symptomatology, which is rated by an interviewer and has 7 subscales. And this is a sample. The striking, I think, which really much fits with the existing literature. If you are asked, have you ever experienced any psychiatric problems, or have you ever seen anyone for psychological concerns, 25%, 26% of our sample reported that they had. And this is compared to 11% of controls. So in terms of CARMS results, we see the mean scores were higher for total psychopathology, all the subscales except emotional disturbance. And when we looked at sort of the high risk kind of band, so looking at the CARM scores equal or above the 90th percentile score for controls, we saw that preterm individuals were more likely to score on a positive symptom scale, cognitive symptoms, and negative symptoms. And I think the obvious question was then, is increased risk of psychopathology associated with structural brain alterations? And this is a kind of a recent paper, which took a lot of time to write for technical reasons. We're looking at altered or cortical gerification and CARMS and IQ in this sample. And the idea is that gerification, which is the folding of the cortical surface, occurs mainly in fetal life. So it's a kind of, could be an important sort of fingerprint of preterm birth for later outcome. So we looked at, we used an LGI, which stands lateral gerification index, the amount of cortex which is buried underneath the surface of the spherical cortical mantle. And we saw that compared with term-born controls, very preterm participants showed reduced gerification, like in 2 clusters, one in the left and one in the right hemispheres, but both had peaks in the insula. But the cluster extended, as you can see, like to the adjacent areas, so the prefrontal, the anterior temporal, medial, and dorsoparatal, and occipital cortices. And then when we looked at the association between LGI and CARM scores, in very preterm group, the associations involved the central and the paracentral lobulus, which corresponded to the primary sensory motor cortices, and the right insula, which we know is specialized in interoception, multimodal and somatosensory processing, and emotional awareness. Whereas the association between LGI and CARM scores in the controls were centered here in the supramarginal gyrus, which is implicated in sort of empathy and social cognition. So to conclude, cortical gerification measured in adult life provides a fingerprint for very preterm birth, with wide alterations persisting as the individual grows older and possibly implicated in functional outcomes. So I think very much kind of to conclude, I'd like to sort of to say that this series of studies showed us that in adult life, individuals who were born very preterm continue to show structural and functional brain alterations in several areas that have been associated with cognitive and mental health problems. And the challenge very much now is, can we use imaging clinically as a biomarker for the identification of younger preterm individuals that are at increased neurodevelopmental risk? And this is very much sort of taking information and like learning lessons from this study. We are now studying much younger individuals and trying to follow them up from childhood, from birth, in fact, up to adult life. I'd like to thank you very much for your attention, and thank my colleague at King's College London that made this work possible. Thank you.
Thank you very much, Chiara. Here I am, I'm back. That was absolutely fascinating. And so I see we have one question posted in the Q&A already. I'm just going to check the chat screen as well. But I am going to use my privilege as chair of the session to ask you a question, if I may. So you touched on the sort of the multiple clinical outcomes that preterm children can sort of and adults can face, but also the possibility of a distinctive preterm phenotype. And this is something we talk about a lot, you know, specifically taking an example like ADHD, for example. Are children, preterm children, vulnerable to ADHD, and that's a valid diagnosis for them, or is there a distinct preterm phenotype which we're labeling as ADHD because we don't have any other label available to try and capture the fact that there's an obvious kind of need and a difference? And I just wondered if you could expand a little more on your thoughts on that, the likelihood that there is a distinctive preterm phenotype, or whether prematurity is a sort of risk factor for other clinical outcomes.
Yes, that's a wonderful question. Thank you, Sue. I think there is this distinct clinical phenotype, and thinking about ADHD. So preterm children, in fact, we published a paper on a younger sample earlier in the year. None of them has ADHD hyperactive type. 100% have inattentive type, which I guess in sort of a sample of, I don't know, 120 children is rather unusual. And also we don't see like the male-female sort of prevalence. So an equal number of girls is likely to be, preterm girls is likely to be diagnosed with ADHD than boys, which I think is quite different from the general, what we see in the general population. And the same thing, it's with other diagnoses. I think the field is really too new to be able to say exactly what and how they differ. But from the study that we've conducted, we see that a lot of individuals have elevated scores, but they do not meet clinical criteria. And I think this is a real kind of problem, because they are distressed, they seek help, but they're not receiving help because they don't meet the diagnosis. So I think it's like gray sort of subthreshold area that is often kind of comorbid as well. And they're not kind of, their problems are not identified, they're not captured. Services specifically for preterm adults or preterm children, like past the age of 2, actually don't exist. And I think there is an urgent, I think as this population is growing, and it will grow in the near future, there should be some sort of special services that are dedicated to their specific issues at the national level.
Yeah, I agree. Okay, so we have some questions being posted. This is terribly exciting. Okay, so the first one from Neelam Sharma says, has there been work done on the nurture aspect of very preterm birth? Preterm birth must be very anxiogenic for parents, and this may affect their parenting and potentially result in anxious children, though it's difficult to measure. So I wonder what your thoughts are on that.
Yes. So yes, very good question. Thank you. There are studies. We are doing it actually now in 9 years old, and parental factors are somehow protective. There are this idea, I think, let's see, what is it called? You probably know. It's like a kind of selective susceptibility models. So the idea is because of I can't think of like better word than kind of fragility of preterm children. They are much more, they're responding very well to kind of positive reinforcement and positive parenting, but also they are more affected by more like negative patterns. So they respond like better and worse at the same time. So I think the environment, it's extremely important for their development, and a lot of parenting strategies that actually diminishing anxiety are being implemented around the world. So I think, but I think it's very tricky because some of the children, and perhaps James is, if it's in the lecture, you can comment on this. They're really sick when they're born, and they are in neonatal intensive care for a prolonged period of time. So the anxious response is also like a normal response to kind of a clinically unstable situation.
Yes, absolutely. I mean, it's very important that we don't use kind of correct or incorrect kind of language about this, right? Like, as you say, that's a normal response and an appropriate response.
Exactly.
Yeah. Okay, so here's a question from my colleague Heather Wally, who says, do we know the mechanisms? She's thinking specifically about how much do preterm adult brains differ in relation to term but low birth weight individuals? So is that contrast, I guess, between preterm birth and low birth weight valuable in elucidating some of the mechanisms of the effects that you've talked about?
It's very difficult to disentangle from the existing studies what is the effect of low birth weight and the effect of prematurity, because a lot of children who are born preterm are also low birth weight, and the other way around. One thing that we have done in a younger sample, which was large enough to be able to differentiate between preterm babies who had intrauterine growth restriction and those who didn't. So this is like different. It's not kind of low birth weight, like specific kind of growth patterns, but there were like really large differences. This is unpublished data in terms of brain volume at term equivalent age, with the limbic system particularly affected. So the preterm children with intrauterine growth restriction had lower volume of extensive sort of limbic areas compared to preterm children that were born appropriate for gestational age.
Fantastic. So here's to our next one. This is James Boardman, who we both know. James says, congratulations for sustaining the cohort over decades. And he asks, have you found any evidence from imaging studies that postnatal factors explain variation, e.g. wealth, parenting, family experience, others?
So in the sample that I've discussed, we unfortunately haven't collected. So we haven't collected an extensive, I think, kind of environmental variables that will be sufficient for us to address these questions. The study was initiated in a pediatrics department and then was transferred to a psychiatry department. They were like very kind of different interests. And I think with the knowledge that we have now, in younger samples, we are collecting really a lot of parental and sort of social demographic data that will allow us to address these questions.
So that's the last question that we've got posted. So I'm going to ask another one and encourage anyone listening to add their questions to the Q&A or the chat if you would like to. So on that note, and I'm sure that you may feel it's a bit early to be able to answer this question, but I'm going to push you a little bit anyway. If you were able to implement some kind of intervention or support for preterm children now, if you were given, you know, government funding to do whatever you would like, what sort of time point would you go in at, and what kind of intervention would you be thinking about applying to head off some of these difficult outcomes that you've talked about?
So yes, I've thought about this a lot. So what I would like to do, I would like to create an intervention and deliver an intervention during the preschool years, because so around the age of 4, because a lot of problems become manifest when a child enters a more complex social environment, which is school. So sort of build resilience before school entry to prevent a lot of problems from escalating. And I would target specifically cognitive control and emotion regulation, which could be, I think, beneficial both for mental health, but also for learning. And one of the main problems of preterm children, like without ADHD, is attention, sustained attention. So I think perhaps targeting kind of core components that can help and support the child through the early school years could be very beneficial in the long term.
And so would you trace a pathway then from those early kind of cognitive and attentional differences to those mental health outcomes that we've seen in later life? You know, would you say that that's a consequence of a sort of negative school experience and the challenges, or is it more fundamentally driven by the biology, you know, by the brain structure and so on, or maybe both?
They're both. I think they're absolutely both. And given also the tendency for preterm children to be more reserved and less extroverted than like term-born children, I think it becomes almost like they enter into like some sort of like spiral that, you know, as they grow up, becomes actually worse and worse, because they become sort of more self-isolated, they're learning less attentive, they have less, like a fewer number of friends. And I think that this impacts both their learning and, of course, their mental health.
Okay, so here we have a question from John Ferguson, who says, thank you for a very interesting talk. Having had a very premature birth myself, I recognize the phenotype of inattentive, not hyperactive, internalizing in my own childhood. I was a complete space cadet. I had always considered this a consequence of spending the first weeks of my life in the relative bliss of an incubator, where all my needs were met, untroubled by external influences. Is there any way of controlling for time spent in incubators to dissect the lack of in utero maturation as opposed to the lack of early maternal nurturing?
Yes, that's an important point. So all the studies we are conducting are accounting for like the length of stay in the neonatal intensive care unit, and they're really like trying to eliminate or account for the effect of a clinical risk. So, you know, if a child or if a baby had kind of invasive procedures, these are accounted for in the analysis. So we are actually looking at or trying to isolate the effect of preterm birth as opposed to clinical risk. As in terms of, yes, how, I think there are some studies, and in fact, in terms of like maternal potentially protective factors that have been looking at the effect, combined effect of clinical risk and environmental sort of protection. I believe Stephen Miller in Toronto, they've done a couple of studies on this, and I'm happy to send the person who asked the question like a reference. That is not what we are doing, but I know some groups, more enlightened groups, and perhaps James is doing it too, but we are not so far.
Okay, great. Okay, so we've got one more question here, and it's a two-parter, and I think we'll probably make it the last one as we approach 5 o'clock. So this person says, hi, thank you for a wonderful talk. I was intrigued about the ideas of increased maturation in subcortical and medial temporal regions, and he has a couple of questions about this. So the first is, do you have any thoughts as to why increased maturation appears to be region specific, or what the underlying mechanisms are driving this maturation? And then the second question is, are there any studies that demonstrate increased maturation earlier in age?
Yeah, so in terms of mechanisms, we don't have any idea. This is like an observational study. Now, in terms of earlier studies, yes, there are studies looking at kind of brain maturation in preterm children, and specifically, what they've been looking for, delayed maturational patterns. So up to kind of early adolescence, children seem to be lagging behind in terms of maturation, which I think in future studies, looking at a much kind of wider age range, it will be really possible to test this retrogenesis hypothesis, because then if the regions who are delaying maturations are the same that are showing accelerated patterns, then we'll be really able to say we're kind of seeing this inverted U shape. Having said this, something that I always think about, if you're looking at the curve, what is accelerated aging could actually be reflected, mirrored on the opposite side of the curve. So without having proper longitudinal data, you are unable to really sort of state whether the development stopped at a certain point or peaked and then declined. So I think further studies will be able to address this directly. At the moment, all we're doing, we are looking for differences between chronological and predicted age, which of course is based on normative data. So if you think the whole kind of development is different, it's like shifted, then we don't have the parameters against which to kind of measure it.
Yeah, and that was something that I was really pleased to hear you emphasize in your talk, because I think it's not emphasized enough, which is that, the process of disrupted development means that we can't assume the same structure function mapping as we would in other children. And so, I think that's something that can be forgotten sometimes in these kinds of investigations.
Absolutely. Yeah. That's why we think it's important, increasingly important, to look at function and structure in combination.
Well, I think we should draw to a close. I see there are a couple of questions we haven't been able to answer, but I'm happy to pass those on and make connections. So please do
get in touch with
me. Thank you, Chiara. That would be much appreciated. We can't really do a round of applause, but I will clap on behalf of everyone watching.
Thank you so much.
And thank you so much for going for this kind of remote option. We really appreciate it, and wishing you every success with this brilliant work that you're doing, that's so important for the group that we're studying.
Thank you, and thank you again for inviting me.
Thank you, Chiara. Bye-bye.
Bye-bye.